Dear students, our lecture today is about complement system. Here are some key definitions will be mentioned during the lectures. Here also, learning objectives of the lectures. Define the complement system. What is the complement system? Mention the three pathways of complement system. Illustrate the initiators and initiation steps of each pathway. Explain steps of complement activation. Mention the biological activities of complement system. Acute phase proteins increase in response to infection. Now, what is the complement system? Complement comprises approximately 30 circulating and membrane-expressed protein synthesized in the liver and by cells involved in the inflammatory response. Named for some of the earliest observations of its activity. A heat sensitive. Now think about that. What do I mean by heat sensitive? That means that the complement is sensitive for heating. The heating of serum containing complement will destroy the activity of complement. It's a heat-sensitive material in serum that complemented the ability of antibody to kill bacteria. It plays a major role in defense against many infectious organisms as part of both the innate and antibody-mediated adaptive immune responses. There are three pathways of complement system: classical pathway, alternative pathway, and lectin pathway. Now, the initiator activators of classical pathway. Antigen-antibody complexes. That means the classical pathway, complement pathway, is a major effector mechanism of the adaptive immune response and leads to elimination of pathogen. Second, aggregated immunoglobulins. C-reactive protein. C-reactive protein is a protein made by liver, its levels in blood increasing during inflammation. The C-reactive protein test measures the amount of C-reactive protein in blood to detect inflammation due to acute conditions or to monitor the severity of disease in chronic conditions. Also, beta amyloid found in Alzheimer's disease plaques. Beta amyloid is big peptides of 36 to 43 amino acids that are crucially involved in Alzheimer's disease as the main component of the amyloid plaques found in the brains of people with Alzheimer's disease. Also, some viruses can activate the classical pathway, including HIV-1. Necrotic cells also activate the classical pathways. Now, is there any difference between IgG and IgM in activation of complement? Think about it. And let us discuss about that after that in Telegram or Let, let's think about it. Okay? The initiation of classical pathway. Here, as you see, in the pathogen have pumps, as we mentioned. What is the pumps in the previous lectures? Pumps on the pathogen will bind with antibody. Pumps as an antigen will bind with antibody. For example, IgG or maybe IgM, and that IgG bind with pumps as antigen on the pathogen. This will called antigen antibody complex. Antigen antibody complex will activate C1q protein of the complement. Activation of C1q will activate C4 and C2, followed by cleavage of C4 into C4a and C4b. Also, C2 will be activated and cleaved into C2b and C2a. C2a and C4b will be the C3 converters. That means this complex C4b to 2b will activate C3, leading to split C3 into C3a and C3b. The initiators, activators of alternative pathway, any foreign substance, lipopolysaccharide of bacterial cell wall, just like gram-negative endotoxins, cell wall of some yeasts, cobravenon factor, poison of type of snakes called cobra, cobravenon factor is the non-toxic complement activating component of cobravenon. It's like naturally occurring C3b. Cobravenon factor forms a complex or convertase with complement component factor B and factor D in the alternative pathway. Also, some viruses will activate the alternative pathway, aggregated immunoglobulins, necrotic cells. All these activators activate the alternative pathway. The initiation of alternative pathway. C3 in alternative pathway, C3 undergoes spontaneous hydrolysis under the effect of activators or initiators of the pathway. It will be split into C3b and C3a. C3b will bind with factor B. Factor B is a complement component bind with C3b in the presence of magnesium ions. C3b, this complex C3bb will be under the effect of factor D which is normally present in plasma as enzyme, will activate C3b and cleavage of factor B into B A and BB small. C3b, B capital B small, it's a C3 convertase. This C3 convertase will split C3 into C3b and C3a. There is a step, a regulatory step, factor H will regulate the splitting of C3 in that case as a regulatory factor in alternative pathway. The activators and initiation of lectin pathway. Initiation of lectin pathway in the absence of antibody. It's very important to remember that lectin pathway, there is no antibody present in that case. Initiation of lectin pathway by MBL. MBL is mono-binding lectin proteins found in circulation bounded with proteases enzyme, known as MASPs. The MASP is mannose-associated serine proteases. We call MASPs. These MASPs are enzymes, proteases enzyme, that found in circulation bounded with MBL and E. coli, involved in lectin pathway of complement system and responsible for cleaving C4 and C2 into fragments to form C3 convertase, activated when MBL bound to BAMs. Let us remember that lectin pathway is part of the innate immune defenses and is involved in the rapid response to pathogen. Here in this diagram, the pathogen with its BAMs bind with MPL, mannose-associated mono-binding lectin and orphicolin, which already bounded with MASPs, the mannose-associated serine proteases. After this complex bind together, activation of MASPs will occur, activation followed by cleavage of C4 and C2, C4 into C4a and C4b, C2 into C2a and C2b. C2a bind with C4b to form a complex structure called C4b2a. It's the C3 convertase which activates splitting of C3 into C3a and C3b. As you see here, the lectin pathway is similar to the classical pathway, but the difference is the initiation will be with MBL and MASP, while in the classical C1q activation. Okay? Here are the final activation of complement, the classical pathway, and as a result of antigen-antibody complex, mono-binding lectin pathway, mono-binding lectin bind monos on pathogen service, alternative pathogen services activate the spontaneous cleavage or C3. All these factors activate C3 and leading to C3 split into C3A and C3B. C3B then lead the final steps of complement activation by activation of C5 as C5 convertase, and then C6, C7, C8, and C9 will involved in the final steps of complement, leading to membrane attacked complex through the polymers of C9 and make this mark membrane attack complex on the cell membrane of pathogen in order to rupture of cell membrane of the pathogen. Here are the formation of membrane attacked complex by C5b, C6, C7, C8, and C9 polymerase are leading to cell lysis of microbes. The biological activities of complement cell lysis through C5b to C9 through the activation of and leading to MAC formation, membrane attacked complex, or on the surface of pathogen. Also clearance of immune complexes by C3b through RBCs, B cell activation, mast cell degradations through degranulation, through C3a and C5a, leading to activation of mast cell to release its mediators, just like histamine and other mediators, and also chemotaxis, C5a as a chemotaxis for neutrophil, and opsonization as C3b on the surface of microbe to lead to phagocytosis. Opsonization and phagocytosis. Here the complement each phagocytic cell have complement receptors for complement on its surface. When this C3b, C4b bind on the bacterium surface as an opsonine, phagocytic cell will recognize the receptor through its receptor for complement and bind with this opsonine and leading to phagocytosis. Here the classical pathway through leading alternative pathway and leading to many effectors, just like opsonine, anaphylaxis, and leading to mast cell degranulation, leading to release of histamine, and neutrophil for increased permeability of vessel, cell lysis, MAC formation, many effector function of complement. Here, acute phase protein increase in response to infection like C-reactive protein, mannose binding lectin, fixed complement, opsonizes, and C3, C9, factor B increased complement function in that case. Thank you very much.